Okay, this is the genetic series. Now, welcome students to this particular topic. Today, we'll be looking at Module 15, and the title of this module is Pedigree Analysis. So, what is a pedigree? A pedigree is simply a record of the ancestry of an animal. Essentially, it is a record that shows all the ancestors of the animals going back as far as possible, through which we can trace its parentage to the grandparents, to the grand-grandparents, and so on and so forth. A pedigree also is a diagram which shows family relationships, and in these relationships we use symbols to represent people, and we use lines to represent genetic relationships. So pedigree diagrams makes it easy for us to visualize relationships within families, particularly in a large extended family. In a pedigree, charts or analysis, people are usually represented by symbols as earlier mentioned, and such symbols are: a normal male is usually represented by an unshaded square, and a normal female is represented by an unshaded cycle. So the shaded square represents an affected male, and a shaded cycle represents an affected female. This shape is used to represent a situation whereby the sex of the animal is unknown. A line joining a female symbol to a male symbol signifies mating. In this case, this is a mating between an unaffected male and an unaffected female. When the lines are true, as is the case here, this is showing a mating that is very close, what we call consanguineous mating. For example, seed matings between brothers and sisters, or between parents and their children. A normal square and a normal cycle that is crossed, as shown here, signifies that that person is already deceased but was unaffected. But if it was shaded, if the square and the cycles are shaded and then crossed, it usually means that the person was affected by a particular disease and the person is deceased. So when we have this kind of shape, is signifying that this individual was a stillbirth or an abortion had occurred. And when we have this kind of symbol, it signifies that the individual did not give birth to children. So pedigrees are often used to determine the mode of inheritance of genetic diseases. If we want to know whether the cause of the genetic disease is due to a dominant gene or allele, or is due to a recessive gene or an allele. So this is a typical family pedigree. This is the first generation. The father married to an affected mother. They gave birth to 3 children, this, and this. So this married an unaffected, and married an unaffected man. So the disease died or was kept in a recessive state within this family. It's obvious that this individual is a carrier. This person, the male, was affected by the disease and married an unaffected person. So they gave birth to 2 individuals, male and female, that are both affected. So it's possible that this person is a carrier and married to somebody who is affected by a particular disease. Okay, and that's why the children are both affected. Here also similarity. Here we have a female that married a man that is unaffected, and half of their children, one is affected while the other one is not affected. We'll see as we go ahead why these kind of situations occur where we have half the children affected, where we have both the children, male and female, being affected. So this is still another example showing us the typical pedigree. So we have the first family. This is the overall. We're tracing it to these people. So here we see that this is an affected male married to a female that is now unaffected but is now deceased. They gave birth to 3 children. 2 of them are affected. 1 male who is now deceased. 2 females. 1 is affected, and this one is not affected. The one that is affected married an unaffected man and transmitted the disease to 2 of the children, while 1 is not affected in the 3rd generation. So, pedigrees actually help us in uncovering the patterns of inheritance in farm animals. And how is this? We want to take a look at some typical examples of an autosomal recessive disorder in animals. Now, in a situation of an autosomal recessive disorder, one of the things that we need to note is that the affected person or phenotype of a recessive disorder is determined by the homozygous or homozygosity for a recessive allele. In other words, if the condition is a recessive one, autosomal recessive one, it can only show when the recessive alleles are in the homozygous state. And then the unaffected phenotype is determined by the corresponding dominant allele. And this unaffected phenotype is because the dominant allele is able to prevent the recessive allele from showing up. And so the person is not affected by the autosomal recessive disorder. So an example of an autosomal recessive disorder is phenylketonuria, abbreviated as PKU, is an example, and is caused by homozygosity of recessive alleles. PKU is determined by an allele that we can represent by small p because in the recessive state, and the normal one is capital P. So therefore, sufferers of this disease, we have the genotype small p, while those who are unaffected will either have capital P, or capital P and small p. Therefore, what patterns in a pedigree will reveal such an inheritance? What do we see in a population that will show us that there is the presence of an autosomal recessive disorder? There are 2 key points that should be noted, and these are that generally the disease will appear in the phenotype of unaffected parents who are both heterozygous. So this disease will come about when 2 individuals that are heterozygous for PKU marry. In other words, their genotype is capital P and small p. And also, what happens is that the affected progeny will include both males and females equally. So wherever you see a situation whereby a condition, a recessive condition shows in a family, the children, both male or female, are likely to have it. The parents will look normal, but they are carriers and hence will transmit the disease to their offspring. So this is a chart to show that these are 2 heterozygous individuals that are married and they're able to produce a normal person, two carriers and one person affected by the disease, PKU. Albinism occurs in man and in many other livestock animals, and is a typical example of an autosomal recessive disorder. Recall that albinism is a disease in which there is lack of melanin in the skin and in the eyes of the individual, and so they appear paler than normal. This is a chart to show us the case of how albinism can come about. So in the first one here, we have a carrier father who married a non-carrier uh, woman or mother. So if we have this kind of situation, because the disease is a recessive one, the non-carrier gene, the stronger gene, will be able to mask the expression of the carrier gene. So what we obtain here is that 50% of the children will be carriers and 50% of them will be normal. But if both of them were carriers, as in the case of PKU, then we have a situation whereby one of them will be affected, this person is affected, two of them will be carriers, and one will be a non-carrier. So the non-carrier genotype representing the allele for albinism is capital A for the non-carriers. The carrier will be capital A small a, while those with albinism small a. So the pedigree below illustrates the point that affected children are born to unaffected parents. So we can see from here that the parents were not affected, but they most likely were carriers, and the children here are both affected. So let's now take a look at autosomal dominant disorders. In this situation, that is autosomal dominant disorders, the normal allele is actually the recessive one, and the abnormal allele is the one that is carrying the dominant gene or the alleles. This is a complete reversal of the autosomal recessive disorder. So a typical example of this is the rare autosomal dominant phenotype is a disease condition called achondroplasia, which is a type of dwarfism found in animals. So in this disease condition, people with normal status are genetically small d, the recessive individuals, and the dwarf phenotype will be either capital D, small d, or capital D and capital D. And the belief is that in the capital D individuals, the two doses of the D allele will produce such a severe effect that this genotype is lethal, and in most cases, they do not survive. So if you're looking at the classical Mendelian ratio, you may end up having a ratio of 2:1 as against 3:1. So if this is true, then all achondroplastics or those affected by the disease of achondroplasia are heterozygotes, as the dominant homozygote is not expected to actually survive. So how do we identify autosomal dominant disorders? One is that the phenotype tends to appear in every generation of the pedigree. In every generation of the pedigree, you are likely to meet an individual that is affected by the autosomal dominant disorder. The second point is that the affected fathers and mothers transmit this phenotype to both their sons and also to their daughters. So these are the two key features to look out for when we suspect a case of autosomal dominant disorder. So this is a classic example of a pedigree showing an autosomal dominant disorder. You can see that the disease appears in this generation, is also appearing in this generation. That it is not appearing in this generation is because both parents here were not affected by the disease. We can also see that it is also affecting both the male and also the female progenies. Another example of an autosomal dominant disorder is Huntington's disease. Now, in Huntington's disease, which is symbolized by neural degeneration, leading to convulsions and premature deaths in animals, the key thing also about Huntington's disease is that it's a late onset disease, meaning that the symptoms most likely will not appear very early in the life of the person or the animal until after the person has begun to have children. That is when we will see the disease appearing in the progeny. So each child of a carrier of Huntington's disease abnormal allele, we have a 50% chance of inheriting the allele and also the associated disease condition. Let's now look at X-linked recessive disorders. So X-linked recessive disorders are sex-linked disorders, and this means that they are carried or show more likely in one sex than in the other. Now, few phenotypes determined by alleles on the differential region of the X chromosome are related to sex determination. So phenotypes with X-linked recessive inheritance typically show the following patterns in pedigree. How do we note them when they appear in pedigrees? 1. More males than females show the condition. And the reason for this is because a female showing the phenotype can only result from a mating in which both the dam and the sire bear the allele. For example, if there's a mating between a female with X capital A and X small a, and then a male with X small a and Y nothing, because these usually do not carry alleles that will counteract the effect of the opposite. Then we could have a situation of an XX small a, and this person will be affected by the disease. But mostly, sorry, if you have an X and Y, because there's nothing to counteract the effect of this, then the male child will have the disease. So a male with a phenotype can only be produced when only the mother carries the allele. If the recessive allele is very rare, almost all individuals showing the phenotypes will be male. The second point we should look at is that none of the progenies of an affected male are affected by the condition, but all his daughters must be heterozygous carriers. Why? Because the females must have received one of their X chromosomes with the affected allele from their parents. So half the sons born to these carrier daughters, however, will be affected for the same reason that the person they marry, the Y chromosome doesn't have an allele to counteract the effect of what is coming from the mother. So this is an example of autosomal recessive condition. So you can see a carrier father and also a carrier mother. So when they give birth to 4 children, one of the males will be unaffected. The daughter, one will be a carrier, one of the son will also be a carrier, and one of the daughter will be affected by the condition. So this is the same cross shown here. This is the mother for this condition. We represent the gametes by capital C and small c, and this is for the father. So this is the unaffected individual. These are the 2 carriers, and then this is the one that is affected. This particular condition is known as cystic fibrosis, And in this cross, you see that 75% of the individual, these three, will not express cystic fibrosis, even though these two are carriers. And then 25% will express the disease condition. So let us now determine if the pedigrees we're going to show after this slide can be for a trait that is X-linked recessive. We're going to use the following designation: that X capital A represents a normal situation, X small a represents the trait, that's a genetic disease or an abnormality, while Y, as we all know, is the Y chromosome, and this can only appear in the males. So you are asked to write the genotype of each individual next to the symbol. And the question is this pedigree possible for an X-linked recessive trait? You have to give reasons why you think so. So next, we have to also write the genotype next to the symbol for each person in the pedigree below. Again, is this pedigree possible for an X-linked recessive trait? Give reason to your answer. So what can you also conclude about the male children of mothers that are affected with an X-linked recessive trait? Cycle the correct answer. So is it that if the mother has an X-linked recessive trait, the male children will not have the trait? Or if the mother has an X-linked recessive trait, then the male children might or might not have the trait? And C, if the mother has an X-linked recessive trait, all of the male children will have the trait. So go through the material again and give answers to all these questions. So you also to write the genotype next to the symbol for each person in the pedigree below. And having done that, is it possible that this pedigree is for an X-linked recessive trait? Give reason to your answer. This is yet another pedigree. You also to write the genotype next to the symbol for each person in the pedigree below. So we've come to the end of this module on pedigree analysis. I hope we have learned much about what a pedigree is and the reason for keeping such pedigrees. One of them is to help us in checking the movement of letter alleles in families of animals, even in human beings. And it also helps us to link up our genetic relationships onto our forebears. So thank you for listening. Don't forget to like and to pass a comment. Be interested in reading from you. Kindly share this with your friends, and lastly, don't forget to subscribe to the channel. Have a great day.